Our next speaker is Dr. Aram Mangasarian. Okay, got it right. The CEO of TME Pharma, a clinical stage biotech company listed on the Euronext Growth Paris exchange under the ticker ALTME with a market cap of about 7.2 million euros. TME is developing therapies that target the tumor microenvironment, a major barrier to cancer treatment success. Their lead program, NeauxA12, is a first-in-class RNA aptor that blocks key chemokine signals, boosting immune infiltration and preventing tumor repair. In glioblastoma, one of the toughest cancers to treat, NeauxA12, combined with radiotherapy and bevacizumab, double survival versus standard of care, hitting a median overall survival of 19.9 months. A randomized phase 2 trial is ready to launch with regulatory clearance in place. The company is also preparing to spin out a NeauxE36 for use in ophthalmology, aiming to unlock further value. Aram, we're excited to hear more about it. Thank you.
Thank you, Eric, and thank you all of you for joining me here today, this morning. I hope you're having a great conference. I'm very happy to tell you about our exciting work in the glioblastoma space here today. As Eric mentioned, we're listed, so there'll be some forward-looking statements. I want to thank Chad and the whole CAGLA team for putting this conference together. I think it's a great initiative and a really exciting conference, and there should be more like these. Also, LifeSci, Eric, thank you for reaching out to your colleagues in Europe that I work with, encouraging us to come here. I think it's been a worthwhile trip and a really interesting time. But let me get into the tumor microenvironment. So this is, of course, these are the healthy tissues that the cancers hijack and use to their own ends, and we're able to target them. This is really important in many solid tumors where there's no good treatments today. Our lead asset is called NeauxA12. We're developing that in multiple combinations. The most exciting data we have to date is in the space of glioblastoma. And I'll take you through that. But in the worst frontline population, which are chemo-refractory patients where the surgeons cannot remove the tumor, we're able to double survival time based on a comparison to a historical cohort. We see radiographic responses in all the patients. We see durable MRANO responses in 5 out of 6 patients, and half of them get complete or near complete radiographic responses. So this is not something you see in this disease. From an investment point of view, glioblastoma is rare, but it's significant enough a cancer that we estimate the market between the US and Europe to be around $2.5 billion per year. Pancreas cancer, which is another tumor we're working on in a collaboration, is even bigger. So the big catalysts for us, based on the phase 2 that's just wrapped up, are getting the phase 1/2 that's just wrapped up, or getting the phase 2 that the FDA approved finance and running, either with us or with a partner. So we're working on both financing and partnering strategies, and getting the spin out, which Eric mentioned, of our second drug, which is de-risked, into ophthalmology. So we've been beaten up like a lot of biotechs, but we've got a committed investor base who wants to see this drug through and get it to patients. We've cleaned up the capital structure of all the toxic instruments we've had to use in the past years to keep the company going. This technology and this approach to the TME is very exciting, and it's what has allowed us to recruit folks like Ulf, who is our chief medical officer, who did some of the work on the first pancreas cancer therapies that were approved while he was at Lilly, and then he moved to Celgene and worked for a number of years there on solid tumor applications. And on our board, Lee Shallop, who is US-based and one of the few folks to get a high-grade glioma drug financed and developed and it's near approval, and you may have seen that Jazz just acquired Chimerix, which is based on the drug that he developed. This is the pipeline. I'm going to focus on the top line, which is the glioblastoma program. We're combining NOX812 with radiation therapy and anti-VEGF therapy, bevacizumab, the antibody, originally developed by Roche. And that's going to be the focus. I won't talk much at all about the second program, which is in pancreas cancer. We have a collaboration with Merck, or MSD as we call them in Europe, where they supply us pembrolizumab. They did that for our phase 1/2. They've agreed to do it for our next study, which has an approved protocol in the US as well as France and Spain, and then E36 in ophthalmology. So what is the drug? It's an RNA aptamer. So this is RNA, but it's not used as genetic material in this case. It folds like a protein. It's outside the cell that'll bind and neutralize a target. So RNA aptamers are interesting because they have a very large interaction surface. That's much larger than a monoclonal antibody, for example. So that allows them to bind and neutralize multiple domains on a protein at one time. For chemokines, that's important because these are the targets we're going after. We can take out the anchor domain that gives the location information, and we can knock out the signaling domain, which is what allows them to communicate with passing cells. So we completely neutralize these, we detach them, we destroy the gradient, and we block their ability to signal. So there's two approved RNA aptamers already. There's an old VEGF inhibitor called Macugen, and more recently there's a complement inhibitor called Izervay, which was acquired by Astellas recently for about $6 billion. What we bring that's new and different is that we've flipped the stereochemistry of the RNA, so we don't need to do any chemical modification of the backbone to get stability. These are completely resistant to RNA's degradation in the blood, unlike standard RNA aptamers. So, to brain cancer. So this is a space that really has been neglected by the pharma industry for years and years. Glioblastoma, the last drug approved was Temodar, which was back in 2005. There's been a device since then. But in the last few years, a resurgence of interest, a number of targeted therapies for low-grade glioma. So the one developed by Agios, which Servier's taken over. Day One Pharma, again in the US, sold their ex-US rights to another French company, Ipsen. In the high-grade space where we don't have these mutations we can target, you have companies like GSK, which is making glioblastoma one of the focus indications for a PARP inhibitor, and you've got Merck that has just paid $30 million to get an improved version of the standard of care chemotherapeutic agent that's used in glioblastoma, Temodar. So what does it look like for patients? It's pretty grim. The most recent data from the US suggests that if you're diagnosed with this disease, your median or 50th percentile survival is 8 months, so less than a year. And within that whole group, there's a mix of patients, as you'd expect, but more than half of the patients are going to be resistant to standard of care chemotherapy. They'll get no significant benefit. And we have a diagnostic that can predict that. That's the methylation status of the MGMT gene. All right? So if you test that and that patient is unmethylated, that patient is not going to get significant benefit from chemotherapy. If you look at that survival curve there, so it's 4 populations. The red and the dark blue line, that's the survival curve for patients that are unmethylated, the chemotherapy resistant. And you can see red is radiation, dark blue is radiation plus chemotherapy. You'll appreciate that there's effectively no real difference there. Now this is an old study, so that the tail is actually probably non-glioblastoma patients. So you can probably safely ignore that. The chemotherapy-sensitive patients, the MGMT promoter methylated patients, do better on radiotherapy alone than the resistant patients on radiotherapy plus chemo. And if you add chemo there, that's the orange line, you get an even better survival. Now, all the patients I'm going to show you from our trial are chemo-resistant. We did that because it allows us to leave out the chemotherapy and propose a chemotherapy-free regimen to patients. The other thing all our patients have is an absence of gross total resection. So we wanted measurable tumor in every patient, A, to assess tumor response, but B, that means we have a much more homogeneous patient population to begin with in terms of expected survival outcomes. And that's probably the only reason we were able to see statistical significance in such a small trial. So let me take you through the mechanism of action really quickly. One or two bits of preclinical data, which is why we got very excited about this particular disease, and then I'll take you into the clinical data, which is the most interesting, I think. So we combine with standard of care radiotherapy. Now, this is very good at burning out the blood vessels in the targeted zone of the tumor. It kills some of the tumor cells, of course, but it's never curative. That drives hypoxia. HIF-1, which is kind of like an alarm, goes off. That upregulates two secreted factors that drive rebuilding of blood vessels. So one of them I'm sure you know well, VEGF. Right? That's the target of Avastin, the Roche antibody. It's been tested in glioblastoma multiple times, prolongs progression-free survival, but it's never shown a benefit on overall survival. One of the reasons we think that happened is that when you shut that down, vasculogenesis, which is a completely mechanistically distinct process to rebuild blood vessels, took over. And that's actually what we target. Now, the target, the protein we're going after is called CXCL12 or SDF1. And how that works is it sticks actually on the inside of the blood vessels of the tumor, and it pulls in passing bone marrow-derived cells that then rebuild vessels. So you don't need a nearby living vessel as you do in the case of angiogenesis. This is like the process of building a blood vessel in development. Now, we were approached by Martin Brown from Stanford it's been like 10 years ago. And he said, look, I see your target going up in my animal model, glioblastoma after radiation, and my clinical colleagues see it going up in their patients after radiotherapy, and we think that's what's driving relapse. So can you send me some of your drug because I want to test it? And our first reaction to Martin was, listen, that's really nice, but our drug doesn't penetrate the blood-brain barrier, so don't waste your time. And he wrote back and he said, look, I don't think you need to, because your target, I think, is on the inside of these vessels, so it's actually in the circulatory compartment. And if you strip that link, which is what's recruiting those cells into the tumor, I think you're going to have a good response. So we sent him drug, and we were pretty astounded by what came back. And I'm going to take you through one experiment. He'd already established that our drug alone, without the hit from the radiotherapy that drives the hypoxia, was essentially giving no activity. So he's using that here as the negative control. That's the black line on the right, where you're looking at tumor volume. Let me say a couple words about the model. So this is important. So this is a carcinogen-induced spontaneous tumor model in normal rats. So you're actually giving a carcinogen during embryonal development at a particular time and triggering mutations. So you're getting a diversity of mutations that result in brain cancer 100 to 200 days after birth, killing these animals. So you're getting a tumor in the right organ in the presence of the microenvironment with an intact immune system and an intact blood-brain barrier. So in a lot of ways, it's one of the most relevant models. It's a pain in the backside because each experiment takes about a year to run, minimum. So that's why not a lot of people use it. Anyway, so what he showed there is that radiotherapy in red and radiotherapy plus chemotherapy, they'll hold the tumor at bay for a little while, but then the tumor grows and the animals die. But when you added our drug after radiation, all the tumors in all the animals disappeared. below the MRI detection threshold for as long as we kept treating with our drug. Now, they came back in 2 of the 6 animals, but the other 4, they lived for another 2 years, cancer-free. So we had 100% complete response, 2/3 of which was durable. Now, he looked at combining with VEGF. We saw some synergy there, but it was not nearly as dramatic as what we saw with the radiation combination. Although it was definitely there, and we could also prevent the influx of macrophages that come in after hypoxia with our drug. And those are thought to be one of the most immunosuppressive populations in glioblastoma. And if I have time, I'll come back to that later. So we decided to target a trial where we treat patients with glioblastoma first line with our drug plus radiotherapy. And then after we saw that data and did some translational analysis of the tissue, we decided to add another arm with bevacizumab, and that's when things got really exciting. So I'll take you really quickly through the trial design. These are all newly diagnosed patients, all with molecularly confirmed, so IDH wild-type glioblastoma, incomplete surgical resections or biopsy only, so measurable, detectable tumor, unmethylated MGMT promoter, so we knew chemotherapy was not going to be helpful. The median expected survival for this group is around 10 months. So the first group got radiotherapy for 6 weeks, which is the standard course in Germany, plus NeauxCancer. We're giving it by a Smith's medical infusion pump, which is often used for chemotherapy. It's available around the world. The patient comes in once every week. We've now done all the stability and microbial testing to do that once every 2 weeks, although we understand that most clinicians are going to want to change the needle at least every week. 3 different doses, 200, 400, and 600 milligrams per week. We didn't see any increase in toxicity. So at the highest dose, after this translational analysis, where we looked at the baseline tissue and saw that actually VEGF was in an almost completely separate compartment than our target, we added, at the high dose, 6 more patients with anti-VEGF. And this is what we see on a patient-by-patient analysis. So each bar is 1 patient, and you're looking at the best tumor response, either in growth in red or shrinkage in green. On the left, and this is historical, this comes from one of the 6 centers in our trial, the University of Bonn, just to give you a rough benchmark of how these types of patients respond. You can see 10% of the patients had an objective response, 25% had stable disease, so between 50% shrinkage and 25% growth, and the rest had a best response of progressive disease. When we put A12 in plus radiotherapy, we already had a big improvement. 90% of the patients had some shrinkage. 40% of them hit the objective response threshold. But these weren't as deep or as durable as we wanted to see. So we worked with the translational group at Bonn. We then tested this anti-VEGF combination. And we saw radiographic responses in every patient. 5 of the 6 patients had durable mRANO, so clinical plus radiographic responses for more than 6 months. And half of them had either complete or near complete radiographic responses. Now, we know that when you've got VEGF, the MRIs aren't always 100% clear. So we looked to the best of our ability with some other advanced parameters to measure things like blood flow in the tumor, and that correlates nearly perfectly with the responses we're seeing here. And more importantly, in terms of survival, we go from 9.5 months on the left, 12.7 months, NeauxCancer plus radiotherapy, and 19.9 months median in the triple therapy combination. The other thing that gives us confidence that our target and our drug is adding something is that if we look at the half of the patients in this middle group where the target is highest in the baseline, the survival is nearly 16 months. So these are the survival curves. So this was published at ESMO last year. or presented, I should say, by our lead investigator, Dr. Frank Giordano out of Mannheim in Germany. What's really nice with this data is that the triple combination hit statistical significance not only versus the historical standard of care cohort, but also versus our other experimental arm without bevacizumab. So this looks like a very strong signal. So benchmarking is, of course, important and very important for investors. They always want to say, well, that's great, but what does it mean relative to other studies, relative to other agents? There's a few things you got to consider. I won't go through this in detail, but the definition of GBM has changed since 2021, so it's now molecular. The older studies were histopathological. If you look, there's a significant, it's not gigantic, but it's a significant percentage of patients who were non-GBM patients in some of these older studies. You got to look at the marker for chemotherapy resistance, the degree of surgical resection, and patient ethnicity in some cases. So we've been tracking the late-stage agents, and we've looked at the data that's available on them. A lot of them have failed in the meantime since we've begun this tracking. But in terms of the median OS, we're well above all of them. At the bottom, if you look in the survival column, at 12.7 months is the Stupp regimen, which is the current standard of care that's given to basically every patient today. Checkpoint inhibitors have failed. Bevacizumab has failed. A number of other targeted agents have failed. The only other approved approach is the one right below ours, which is at 16.9 months versus our 19.9, which is the tumor treating fields. But the benefit shown in that study in this population was 2 months. And that involves shaving your head every day and wearing the electrodes for 18 hours. So it's a rather burdensome therapy, and it works quite well in the chemotherapy-sensitive population, but like almost all the other therapies, very poorly in this chemotherapy-refractory population. So, of course, the other benchmarks are how do you compare it to other regimens with bevacizumab? On the left, you can see one of the trials we found that was rather close to ours in terms of design. So chemo-resistant patients first line that got radiation, chemotherapy, and bevacizumab at the same time. So effectively, swapping our drug for Temodar. We published 21-month OS data. We were at 50%. Here you can see they're at 20%. Even though that trial had fully resected patients who should live on average 5 to 8 months longer. If we compare to the big bevacizumab trials, where they included not only fully resected patients, but chemotherapy-sensitive patients, we're still beating them, even though we have 2 factors that tell us we should be doing worse. So again, that attests to the strength of the signal we're generating. The safety and tolerability look excellent. You can see here breakdown of the adverse events by attribution of the clinicians. So most were no relation. By far the biggest second factor there was the disease itself, glioblastoma, then radiation and bevacizumab. There were 4 attributed either completely or partially to our drug. The ones that were only attributed were essentially administration site issues. And if you look at the overall profile, I know you can't read this, but we had essentially grade 1 and 2 adverse events other than infections and CNS, which are most likely due to the disease itself. So this has a really mild adverse event and tolerability profile. So we spoke to the FDA last year. They granted us fast track designation just after we opened the IND for a phase 2. You know the Optimus guidelines where they want companies to test multiple doses. They asked us to test 3 different doses of our drug with bevacizumab. So this is the phase 2 we would like to do next. 3 different doses of our drug, 100 patients total, 20 per arm, comparing that to 1 arm without bevacizumab and 1 arm with standard of care. So this is open. We have drug to start. We're currently looking to lock down the financing for this. If we don't hit significance here, we would plan to amend and expand that to a larger trial to make this pivotal. So in terms of commercial protection, so the composition of matter patents on this are old, but we have multiple combination patents, including one filed in 2022 that we are very confident to get commercially relevant protection from. So that'll give us protection into the 2040s. In addition, in case that doesn't work, we of course have orphan drug exclusivity, which we've already been granted in the US and Europe, and is obtainable in Japan and China for glioblastoma. So I'm going to wrap up by saying we've got a drug where we think we have the potential for unprecedented activity in this horrible disease, where we're seeing very high rates of response. We've already seen a statistically significant survival signal. Safety and tolerability are excellent. And this is a commercially relevant target market, even alone, despite the fact that there are multiple additional ways we can expand this, including for things like brain mets, as well as the other solid tumors in different combinations in areas like pancreas cancer. So with that, I will wrap up. Thank you for your attention. Very excited to talk to any of you who want to learn more afterwards.